Hey besties! That's right, we're back with another Chem 116 lab video. This is apparently our last video of the semester. Yay for the end of our mutual suffering. You heard right, folks. You don't have to watch another one of these again. You're welcome.
Anyway, as you can see, we started by making our copper 2 tetraamine solution. We did this by weighing out 0.125 grams of our salt, copper sulfate pentahydrate, and dissolving it in a small amount of water. We then moved to the fume hood and slowly added concentrated aqueous ammonia to the dissolved salt. Finally, we moved this mixture to a 25 milliliter flask and added water to the line. In this clip, you can see that our solution has become cloudy and a lighter shade of blue, meaning a precipitate formed. We fixed this by simply adding 10 additional drops of the aqueous ammonia, still being careful that the meniscus does not go over the line.
We've prepared a series of 3 dilutions from our copper 2 tetraamine stock solution. We diluted each subsequent solution by adding 5 milliliters of the previous solution to a volumetric flask, and adding water until we had 25 milliliters of diluted solution.
It is very important to keep track of measurements when making serial dilutions, so you always know what volume was used for each. Otherwise, your calculations will not be accurate later. Also, always be sure to prime your pipette before use. Remember, flush 3 times with water and 3 times with stock.
Now that we have all of our solutions, we transfer each one to a cuvette. Using a transfer pipette, we make sure we transfer just enough solution that the bottom of the meniscus is level with the marking on the cuvette. We prepared 3 dilutions, so each of those is in a separate cuvette along with our stock solution. We brought our cuvettes to the spec room to use the spectrophotometer. First, we used our blank to make sure the spectrophotometer was zeroed.
Before we placed the cuvette into the machine, we carefully wiped it with a Chem wipe to minimize smudges or debris on the cuvette that could affect our results. We then zeroed the spectrophotometer.
After we zeroed the machine, we used a stock solution to scan across all wavelengths so we could determine the best wavelength to test the remaining dilutions on. This wavelength is the lambda max value. It's the wavelength where the solution has the highest absorbance. By finding this wavelength in our stock solution, we could determine the concentration of stock in our dilutions using the absorbance of the lambda max wavelength. Our lambda max value was 606 nanometers. Once we determined the lambda max, we could scan our remaining samples on that specific wavelength. We wiped the cuvette with Chem wipes, placed the sample inside, and closed the lid. We scanned the sample and wrote down the absorbance before carefully removing the sample and closing the lid again. Here's the data we wrote down on the absorbances of each dilution. After collecting all of this data, we made sure to clean up our station before heading back to the lab to continue analyzing our data. Using the absorbances, we were able to determine the relationship between the absorbance of our substance and the concentration of each dilution. To do this, we first entered the data into Microsoft Excel, and then used the program to graph the curve of the relationship between absorbance and concentration. Here's our graph and the equation of the line. Another important thing about the relationship between concentration and absorbance is that it can also be described using the Beer-Lambert law. The equation for the Beer-Lambert law is shown here. The A stands for the absorbance of the substance, and the C for the concentration. Epsilon here stands for the molar attenuation coefficient, which can be found by calculating the slope of the concentration versus absorbance curve for that substance. L is the path length of the cuvette, which remains the same for our experiment as long as our materials remain constant. We hope you enjoyed this video. If you liked it, you know the drill. Like, comment, subscribe, and ring the little bell to feed our egos. If you feel bad for us, consider giving us pity points. They're a great way to contribute to the local tired college student community. Us. We're the local community, and we're always taking donations. You can also donate coffee and energy drinks as finals are rapidly approaching. Several mental breakdowns are also rapidly approaching, but we try not to think about that. Again, thank you so much for watching. We hope you enjoyed this video as much as we enjoyed doing this lab. Interpret that as you will. Bye! Make good choices!